Hey everybody, and welcome to this lecture on leaves. This unit is going to focus on the internal structures of leaves that are important in the movement of water around the plant and the exchange of carbon dioxide and oxygen in photosynthesis. Knowing what these structures are and what their functions are helps you make educated decisions about managing the propagation environment in ways that optimize plant growth. And importantly, also helps you troubleshoot when things don't go quite as planned. By the end of this unit, you'll be able to describe the purpose of stomata and list the environmental factors that influence the opening and closing of the stomata. So let's start by looking at the outer surface of leaves. This complicated looking diagram on the right shows a cross-section through a typical dicot angiosperm leaf. The outside surface of leaves is protected by a thin waterproof layer called a cuticle, which is composed of a hydrophobic fatty molecule called cutin, and it's also impregnated with plant waxes. The main function of the cuticle is to protect the leaf from desiccation by preventing the evaporation of water through the leaf surface. The cuticle also protects the leaf from harmful, shorter wave ultraviolet radiation. Basically, think of the cuticle as a Gore-Tex rain jacket with sunscreen built in. The thickness of the cuticle varies between species. Plants native to hot, dry climates have a very thick epidermis, which provides a lot of protection from desiccation and harmful ultraviolet rays. Examples of plants with a thick cuticle include plants that you can see in the picture on the right, such as these agaves, prickly pears, and other cacti that are native to the Sonoran Desert in Mexico, Southern California, and Arizona. In contrast, aquatic plants which spend their lives submerged in water tend to have a very thin cuticle, or sometimes no cuticle at all, because drought stress isn't going to be an issue for them. Plants native to shaded forest or woodland ecosystems, such as this fringe cups, which you can find growing in our local coast redwood forests, tend to have thinner cuticles than sun-loving plants, such as our California native manzanitas and South African leucadendrons, which both grow in chaparral-type plant communities with hot, dry summers. Just below the cuticle is the epidermis, which completely surrounds the leaf. It consists of mostly unspecialized cells and is usually just one cell thick. Epidermis cells contain flavonoid pigments which filter out harmful, longer wavelength ultraviolet light in the 320 to 400 nanometer range. Although most of the cells in the epidermis are undifferentiated, the epidermis cells of some plants may be differentiated as trichomes or glands. Trichome is a collective term for epidermal hairs, bristles, prickles, or scales. And in nature, trichomes and glands on leaf and stem surfaces are adaptations which help the plant survive periods of environmental stress, such as drought, heat, or cold, or they might provide protection against leaf-eating predators. A well-known example of a plant with hairy leaves is lamb's ears, Stachys byzantina, whose leaves are covered in soft, silky hairs which make the leaves appear silver, and of course give this plant its common name. Another example of a plant with hairs on its leaves and stems that are more bristly, and definitely nowhere near as nice to touch as lamb's ears, is stinging nettle. Some plants have epidermal cells differentiated as glands, which exude waxes onto the leaf surface. These waxes give the foliage a silvery or whitish powdery bloom that you can wipe off very easily by touching the leaves. Many succulents have this waxy bloom on their leaves, and its white color helps to keep leaves cool by reflecting solar radiation. Leaves with this powdery waxy coating are described as being glaucous, and examples include many of our California native Dudleya and the South African succulent Cotyledon orbiculata, or pig's ear, which you can see in the bottom right here. Other plants have glands that exude aromatic oils, and many of these plants are commercialized by humans for culinary uses, and well-known examples include rosemary, basil, thyme, and oregano. Other plants that exude aromatic oils, such as lavender, are used in the perfume and cosmetic industries and may also have medicinal uses. In propagation, the presence of trichomes or glands on leaf or stem surfaces doesn't usually affect the techniques you use, but you may have to manage the propagation environment a little differently for plants that have hairy leaves. The problem is that the hairs trap water droplets, which for some species is exactly what they're supposed to do in nature. In the greenhouse and nursery, though, the presence of these water droplets provides an ideal vector for waterborne fungal spores to be spread. They can also prevent gaseous exchange from taking place and cause the leaves to rot. So you may need to reduce humidity levels in the propagation environment for hairy plants and pay very close attention to nursery hygiene in order to reduce the risk of losing plants to fungal disease. Let's take a look now at the vascular bundles in leaves. A plant needs a way to transport water, nutrients, and the products of photosynthesis throughout its tissues, and this happens through the vascular bundles. Vascular bundles are distributed throughout the leaves as well as the shoots and roots, and are visible on the external leaf surface as veins. The vascular bundles consist of xylem, phloem, and cambium, and in many plants are surrounded by a bundle sheath. Think of the xylem and phloem as tubes, but with a more complicated structure. The xylem tubes transport water and minerals from the roots to the rest of the plant, and the phloem tubes transport sugars, the products of photosynthesis, throughout the plant. The xylem and phloem are separated by a layer of cambium in dicot species. Cambium is a type of growth tissue that produces new cells, and you'll hear about this meristematic tissue in a later unit. The vascular bundles also provide structural support for the leaf blade. Let's take a look now at another structure on the leaf surface. In order for photosynthesis to take place, plants need to take in carbon dioxide from the atmosphere and get rid of oxygen. This exchange of gases between the plant and the atmosphere happens through small pores in the epidermis and cuticle called stomata. Note that stomata is a plural noun and stoma is the singular noun. The number of stomata on each leaf and their pattern of distribution across the leaf surface differs between species. But as a general rule of thumb, there tend to be more stomata on the lower leaf surface than on the upper surface. And some species don't have any stomata on the upper leaf surface at all. This is because the lower surface is usually more shaded, and therefore the plant won't lose as much water through these shaded stomata when they're open. The stomata are surrounded by two specialized cells called guard cells, which always make me think of two sausages when I see them in diagrams like the ones on the right. Guard cells expand when they're filled with water, and this causes the stomata to open. Conversely, guard cells shrink in response to water loss, and this causes the stomata to close. In C3 and C4 plants, photosynthesis slows down when the stomata are closed because the leaf can't absorb carbon dioxide. Slower photosynthesis means a reduction in plant growth. Therefore, if you're wanting to optimize plant growth, which we always are in propagation and production, the stomata need to stay open for as long as possible. This means making sure that your plants, and this includes cuttings and young seedlings, have sufficient water and aren't drought stressed in any way. The opening and closing of stomata isn't just dependent on the water status of the plant, though. It's a complex interaction between water status of the plant, temperature, light, vapor pressure deficit, and internal carbon dioxide levels. Stomatal responses to these environmental factors varies between species, so there's no single recipe of environmental parameters that works for every crop. And this is part of what makes growing plants so fascinating and sometimes so darn frustrating. Let's take a quick look at stomatal response to high temperatures. Increasing light intensity causes an increase in ambient temperature and leaf temperature. High temperatures cause plants to lose water more quickly through transpiration, and this results initially in the stomata closing as the guard cells shrink in response to the loss of water. If the ambient temperature stays high for a prolonged period of time, though, the plant can become too hot, and there's the risk that the plant will experience heat damage to its cells. Young seedlings and new growth on cuttings are particularly vulnerable to heat damage as they don't yet have a fully developed cuticle, so they have very little protection. Even mature plants that are moved from a more shaded environment to a hot, sunny environment are very vulnerable to heat damage. When temperatures are particularly high, the plant's response is for the stomata to open again to allow evaporative cooling to take place through transpiration. This may seem a counterintuitive response to us, but the alternative for the plant is cell damage if it becomes too hot, and in a worst-case scenario, plant death. Armed with this information, a temporary period of water stress and the consequent slowdown in photosynthesis seem a small price to pay. So let's summarize what you just learned about stomatal function. As a general rule of thumb for C3 and C4 species, stomatal opening is triggered by increasing light and increasing temperatures up to optimum levels, by low carbon dioxide levels inside the leaf, and by a low vapor pressure deficit. Stomatal closing is driven by the reverse: decreasing light, very low or very high temperatures, high internal carbon dioxide levels, and a high vapor pressure deficit. So what does this all mean for us as propagators and growers? Well, it means we need to keep a close eye, a really close eye, on what's going on in the propagation environment and take steps to manage it in ways that optimize growth of our plants. We need to make sure our plants don't become water stressed. We need to prevent the temperature from becoming too high or falling too low. And we need to manipulate the light intensity and light quality. Later in the production cycle, we may also need to control light duration or photoperiod. The structures and equipment we can use to manage the propagation environment will be discussed in the next module. That's the end of this lecture. Take a break now or head back to Canvas to continue with the other units in this module.